Howdy everyone. Today I'm going to be talking about biochemical pathways, but more specifically I'm going to be talking about them in relation to nutrition and the diet of animals, right? And so all good nutritionists are going to make sure that there are carbohydrates, protein, fats, and so many other things within a diet for animals to ensure that their bodies are functioning and it's happy, healthy, right? But on the molecular level, biochemistry plays an entirely important role in digestion, in health, disease, and all of that within animals. And so right now we're going to start with the backbone of this biochemical pathways that are involved. And it's going to be this one right here. And we call this glycolysis. Glycolysis is going to be, again, the backbone and how we generate that ATP that we need in our body for many other pathways to function, to thrive. And so that we take glucose and through numerous factors here, we go and we get to start breaking it down to produce these ATPs, ultimately to get pyruvate. And pyruvate is going to send a catalyst to so many other pathways. And so this is going to be the true focal point of this backbone, pyruvate, because then we're going to take it into acetyl-CoA and go into the Krebs cycle. And so we have that can I keep selling substances for money officer, right? And so we're using that water, we're producing that water, that CO2, and we're getting these very important intermediates right here. These NADH and FADH are very important to other pathways as well. And so we have some ATP coming in here too. But one big thing that I really like about the Krebs cycle is its versatility. We see amino acids here, right? We see them entering in this cycle or being utilized by this cycle. And this is an important factor to note because all proteins, right, are made of amino acids, which I'm going to put as AA. And so we have histine, proline, glutamine, arginine. And then down here with succinyl-CoA, we have isoleucine, valine, and others, right? Over here we have trypsin and phenylalanine, right? Right here with that fumarate, which are important steps in that Krebs cycle, but also offshoot into many other things as well. And I also want to point out something big here, especially to our feedlot gurus and our ruminant nutritionists. Right here, we have propionate. This is where it's going to enter this cycle, the Krebs cycle. And it's, again, a very important factor, as it's a volatile fatty acid that is known to be good at laying down intramuscular fat, and which goes to marbling in the beef industry. But as we go through this Krebs cycle, right, we're actually going to go to the real powerhouse of the biochemical pathway, which is going to be the electron transport chain, where we're going through our different complexes, right, 2, 3, 4, and our special Q and C complexes. And as we're going there, we're pumping out hydrogens, but we're seeing this NADH again, and we're seeing this NAD, FAD, which again are really important. And then we get down here and we have some water and hydrogens. And then boom, we're done, right? Well, let me just ruin that for you. We are not done. We've only just done this part here. And we have so much more to go. But where do we go from here? Well, we're going to start back at malate. And we're going to go to glucogenesis, right? And then we're going to go all the way back up. Back to the start, this glucose, right? So glucogenesis can't fully go back up glycolysis, right? So we can't just do a full reverse here. There's some reversible steps. But there are other irreversible steps that we have highlighted here. And so we have to use malate as a shuttle to oxaloacetate, and then phosphenylpyruvate carboxylase to go back through this irreversible step. And then we follow it back up, but boom, we get to another irreversible. And so we got to actually use fructose 1,6-biphosphatase. And then boom, to our next irreversible right here. The irreversibles are done by stars to get back to give us that glucose. But going over here, we have a glycogenesis where we're actually taking that glucose, right? And we're actually going to store it into glycogen. And so we're using that UTP to UDP, and we're actually going to store that to where if we need to in our bodies in stress or we need that glucose, we can use that from the glycogen stores, right? But now we're going to go back to one of my favorites, the Krebs cycle. And we have so many intermediates here, but as you'll see, I'm just going to give us a little pre-warning. Alpha-ketoglutarate is going to be quite important. But boom, back to pyruvate, that powerhouse. Could go to lactate, right? Lactate's going to then shuttle us to the Cori cycle because our bodies are so sophisticated. And they know that you can't easily go from muscle to liver. You can't just go boom, muscle to liver, right? We actually have to be able to transport it through the blood. And so we have our lactate here. It's going to go through glycolysis. We're going to get that pyruvate. But then we're going to have to go through transamination and get alanine. And that way, that alanine can be shuttled from the muscle through the blood into the liver. And then we do the reverse, the alanine to pyruvate. And that way we can use glucose. But we got some little deamination going on here. And over here we got some alpha-ketoglutarate, right? What did I say was important in that Krebs cycle? And it's going to take us up into a special thing called urea. And that urea is what we're going to talk about here, the urea cycle. So urea in ruminant animals is a non-protein nitrogen source, right? And so it's important. Now, there's a debate on what number you'll go up to. Some people will go up to 1, but a little more conservative based off of my experiences. So I don't like the diet to go above 0.5. Really, I'll say 0.7. But we're not going to go over this, and this is the most extreme in my opinion. But anyways, you've got that amino acid, alpha-keto acid. What did I say? Alpha-keto is going to be important here. And here we go again, alpha-ketoglutarate, right? And boom, we're hopping to the urea cycle, right? And so we're going to take these different intermediates, the CO2, the NHs, the hydrogens, and all of that. We're going to use some energy here, and we're going to go into this cycle, again, using some energy. But we're using energy, right? So we're using ATP. But once we get over here, there's fumarate again, that Krebs cycle being heavily involved. And so if we actually want to take that and travel it all the way back here, now you guys are starting to see why these cycles are so important and interwoven, right? But anyways, we are using ATP, but guess what? We're getting this non-protein nitrogen source, right? Which is crucial in some diets. And so just because we're using ATP, now we want ATP, we want energy, we want glucose in the body, especially for our brain. Glucose and the brain go hand in hand. They love it. The brain is a glucose hog. And so we need ATP, but sometimes we're willing to spend it to get another source out of it. In this case, a non-protein nitrogen source. But we're not done. Let's go back to that backbone, glycolysis. Here we are, right? And then we're going to go to a very, very important part. This is the pentose phosphate pathway, PPP. And it's very important. Again, we're seeing these intermediates that are so important, that are being produced in these pathways, right? You see here, FAD, NAD. And we're taking it to the ribose 5-phosphate. And then we go through multiple pathways that can be interwoven and changed to get this pentose phosphate. But one of the big things from this cycle is the ribose synthesis, right? The ribose synthesis. Now, DNA, what is that? That's deoxyribose, right? RNA, ribose. Boom. Big player in ATP, right? But also, we're going to talk a little more about these guys. So DNA, we all have it. We all know we need it. We all know it's important, right? We learned that very early on. We have adenine with thymine and cytosine with guanine. And then we have uracil when we're talking specifically about this RNA, right? It has a little fun guy, right? But these are all important in DNA and RNA synthesis. We need it for our body. We know it's crucial, right? And so we're taking our ribose, and again, let's connect these together, right? We're taking this ribose right here, and we're bringing it right here, guys. It is crucial that we understand that this isn't just separate pathways that are all acting on their own. These are all interwoven. And so we're going to take that to get our PRPP. And then, boom, guess what? We're taking that and we're putting it to another pathway, right? And it's going to be used to give us that IMP, which is going to be crucial for the DNA synthase. And we're taking it, boom. What did I say again? Boom, boom, boom. Need that, boom. Look at that. We're going all the way back. And then we're going to get this synthase so that we can have our DNA and RNA utilized within the system, right? And so I hope this gives you a little overview of the biochemistry of just mainly looking at the diet here, because this all goes back to what we're feeding, the carbohydrates, the amino acids, the fatty acids, and truly bringing them together and understanding that diet is affected on a molecular level, but also affects the system as a whole. And so I hope I've captured that today. And so thank you all.